Hello everyone, and thank you for attending today's webinar, A Solid Foundation for Simpler and Smarter Panel Design, presented by Bob Balderas. Bob is the Vice President of Biological Sciences at BD Biosciences. He is a distinguished BD Fellow with over 40 years of leadership experience in both academia and the industry sector. Bob serves on various editorial boards and is a board member at FOSIS and a member of the Dean's Council at UCSD School of Biological Sciences. Bob has published in over 75 scientific journal articles and is recognized all over the world as a visionary that helps create academic and industry partnership for the advancement of science. He has been honored with 2 prestigious BD Howe Awards. Bob has a passion on sharing best practices in the areas of immunology, high-dimensional biology, and flow cytometry. He has been part of worldwide educational seminars and tours in the last 20+ years, and we are excited to have him share his recent ideas on panel design with all of you today. If you have any questions during the webcast, please submit them through the Q&A widget of your console. After the presentation, Bob will be answering your questions in a live session moderated by Manisha Sundarajan. We would like to take a moment to congratulate our clinical lab scientists who are celebrating Lab Week this week, and would like to remind you that attending this webinar would provide you 1 C credit for attendance. Before we begin, I wanted to cover a few housekeeping items. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help Engagement tool at the bottom of your screen. A recording of this broadcast will be made available after the presentation. With that, I will now turn it over to Bob.
Hello, everyone. On behalf of all of our BD associates who've worked over the years on so many different technologies and tools, it's my pleasure to provide this presentation today. I'd also like to say hello to many collaborators and friends and colleagues who've also joined. I've seen a long list of all the individuals that are with us today, and for all of you, I'd just like to say hello and thank you for all the discussions over the years. Thanks for all those papers that we've shared on and the discussions that have been rich in our field. But I'd also like to preface a few things before we begin, because this topic is one that is dearly important to all of us in the field of immunology. For many years, we've watched carefully at BD to learn more about the needs of individuals in this field as it relates to getting great data from the instrumentation and the reagents that are built, not only by BD, but by all the different companies. And we keep track of that information. And what's interesting over time is that issues and questions about panel design, compensation, spread, new colors just continue to come in. And it never seems to change. It's just a constant number of those types of questions that come in. So we know that this is a very, very important topic. So today, we're not going to go through just how that exact panel is built, because because you have all different types of areas of biological focus, but what's underlying in every single panel are fundamentals. And if we use these fundamentals, then we can begin a more rational approach to what we do. It allows us an opportunity to start a panel in a certain way, to build a good panel with hopefully not too many iterations. But these fundamentals can be embedded in a way, if we learn them well, to help us correct a panel where there's something that's just not allowing us to get the most out of a panel, to see the greatest amount of biology. And I know there's many individuals on this line today that have great skills. And it's a mix of art and it's a mix of data and experience that allow. And oftentimes those experiences that we have are actually hard to share to others because we just know a lot about all of those colors that are embedded in the types of panels that we work on today. So I'm going to take everybody through some structure that if we go home today with a knowledge of these, or just maybe just to realize that they're there and to see the types of tools that are available and see those types of approaches, I think that everyone will be able to look at their panel in a different way, maybe make it a little bit better, but most importantly, to get the most and the greatest biology from everything that we do. One other thing that I would like to point out is that there are a number of individuals in our company, individuals on this slide, that many of you know. You've interfaced with them by telephone or in the lab. So to Ravi and Andrew, Stuart, Eric and Monisha, as well as Marianne. These are individuals that help contribute to this presentation because it was an opportunity for us to work together to challenge ourselves and to make sure that the information in this presentation is of value. And it took a considerable amount of time to bring it all together, because what we wanted to do for this Harmony program is to take a bigger step beyond what we did about 5 years ago with the Horizon program. That gave us the first glimpse of many new colors and how we could integrate those colors into great panel design. And we looked at it from many different ways, but today what we've done is taken a lot of those learnings. We've brought those together with all of those discussions we've had with scientists all over the world. And we've tried to put them into a framework that will help, all individuals build a better panel. As we begin today, we'll always look at the panel that we just made, which in many ways, or oftentimes, is suboptimal. Meaning, I think if we looked at our panel, we might be able to say, I wish it could have been a little better on a given, specificity, and to pull a different type of population out. So today, as we look at this slide, we've made a series of changes to obtain a more optimized panel. But what's interesting here is that we just use the fundamentals to edit it. And it's like an engineer. An engineer always takes things apart. And they take them apart because they want to know how it works inside. And once they do, they can fix it very, very easily. So when we look at a panel, if there's some structure around it, it allows us to make iterative changes to be able to get it to a better foundation. Because what we want to do is save time. We'd like to save the cost of antibodies so that we don't have to buy many of them to go through these iterations. And the more time that we spend up front before we go to purchase is a critical step in what we do. And the more fundamentals that we use within those steps prior to obtaining those antibodies, it will pay off. And sometimes it's even better to actually let another person review your panel so that the mistakes aren't made, because sometimes those panels are just too close to all of us. Now, over the years, there's been many learnings. And again, as I said, many of these learnings have come from our conversations, our visits, our meetings that we go to, where we share. But some of these things are critical to how we begin that panel. And because remember, like a chess game, the first move that we have can dictate a good game. And if we make the wrong one at the beginning, it doesn't take the other individual to beat us in that game. So it's clear that we all seem to start designing panels in different ways, and truly that's okay. In our steps through this process, though, many of us do take the time to draw out a biological tree that lays our foundation. It tells us what we're looking at, and it defines what types of colors that we might want to use. And it also shows us, when we build these, that once the tree branches separate, that something we do to one side is not going to impact the other, because the colors don't jump across from one cell to another. We also know that a real foundation of understanding of receptor and protein expression enhances, the success of a panel. And the question there is, where do we get all of that information? And today I'm going to share you one published site and one that is new to be published in the mouse world, because we haven't forgot of the importance of all the mouse experiments, and we've never had some of the data that you're going to see today. I think for all of us too, we recognize that the first color on each of our lasers is really our best. And that goes for cross-laser excitation and spread and spillover. And we're going to go through this because there's a story on every laser line. One of the other important things of learnings that we've had too is that, the last color on the laser lines are very critical colors too. And again, it's part of that story that's unique to every laser line. Well, it goes without saying that sample prep is everything, and the choice of the protocols that we use, the age of the sample that we have, whether it's isolated PBMCs, if it's blood, or if the cells come from lavage, all of that is under your control. And we all know that the greatest flow comes from a great sample. So that's one thing that really is out of our control a little bit in some ways for collaborations, but in many other ways we have full control over that. So our dedication to our protocols are critically important. But other things that have come over time and learnings have a lot to do with how we introduce bias into a panel. And that can be from several perspectives. Number one, it could be we might be looking at intracellular proteins. And as what we do know in our field is that not all colors work in intracellular applications. So that being the case, we have to save those colors to put them on the intracellular ones first, and then we have to work without some of those good colors for all of the other cell surface receptors. Now, bias also can come in if we're looking at activation markers. And if within our panel, we might be looking at basal levels and we're looking at activation levels following some type of challenge, where the numbers of receptors can go from 5,000 receptors to 100,000 receptors. And if we pick the wrong color for that, then it can be a problem in our design of our panel. One of the other learnings, too, that's critically important, and one that we have to be careful with, though, is that the antibody titration is a critical parameter. And this antigen density that's there, as well as the antibody titration from saturation, let's say, can be used to actually help drive down the cost of a panel. Depends upon where it is in the panel itself. So if we're looking at a gating population, such as CD4 or CD8 or CD19, where in some cases we could have a 4 to 5 to 6 log shift between the negative and the positive, well, we know we don't need that. We can dilute that antibody and work with a smaller mean channel shift. But there's an impact when we do that. It actually can be a positive one because we're reducing the amount of color or that color on a specific cell. But we're not saying dilute every antibody. For some, we can do it based upon what our goal is, and for others, saturation is critically important. So these are part of learnings, and there's many more. And we can appreciate all of those, and that comes from experience and things that we do every single day in this field as we communicate with new users or individuals that are looking at all of their experiments. Now, one of the other learnings that's not on here, though, is, and I'd like to give recognition to many people that too are on this call today, and because I'm not going to address the core directors and the core managers and the core scientists that actually get many of those boxes ready every day, that set those instruments up so that individuals can come in and do their work, that filter those boxes to meet the expectations of end users or to provide more information so that it can make their panels and their biology better. Without those individuals in this field, I think it would be a completely different world out there. And we should take advantage of those, even if that flow cytometer that we're using is even in our own labs and not in a core, finding those individuals that have such great experience, they can help you in your labs too. So that's a critical component of great panel design, and because at the end of the day, if the instrument's not set up correctly, then it's hard to get the greatest information out of any given box. We step forward through this deck. We're going to look at a number of different topics, and the first one is that fluorochromes themselves reveal different levels of biology, and how we match those antibodies to a color is a critical component of being able to have a fantastic analytical and sorting panel. This first slide in this section is about color choice and receptor density. And Jake at BD in our San Diego facility built a slide for me that he and his team worked on that gives us a chance to look in lots of different dimensions. I only chose 3 of about 20 different experiments that were done there. And I chose a couple of them because these tell a great story. So we look at the expression of CD56 and CD94. And these are unique types of receptors. And in some cases, as we look for CD94, it's a secondary receptor. It's one where there's a continuum of expression. And if we look at the brilliant blue 700 in the middle, we can see three defined populations there. But if we look over at PerCP Cy 5.5, we don't see that. And if we look over at BV421, we certainly can see that there are three, but there's some spread into that APC channel. So how we look at the individual color and then how we look at that color as it spreads into other channels is critically important for us to be able to get, again, the best data out of any given experiment. So color choice and receptor density are critical components. Several years ago, Mario Roeder and Yolanda Menke, published a chapter in a book that was just a critical one for how the information can be used in panel design. And they classified receptors in different ways, in primary receptor, secondary, and tertiary. Those primary ones, if every antibody specificity was primary, flow would be considerably easy to do if it was like a CD3 and a CD4. But many of the specificities, like CD45RA, exhibit a secondary receptor type, such that there's a continuum of expression, meaning there are many different subsets within CD45RA. And thus, our ability to spread those out is dependent upon the color that we use. And then the tertiary ones that are very low expressed in many cases or have variable expression, such as in activation, are another challenge for us. And Mario and Yolanda built this table that shows multiple different colors against multiple specificities and then categorize them in a way to know if they're primary, secondary, or tertiary. And it's always intrigued me that those three types of receptors, if we look at them from that way, help us in defining what colors, that we do use. Another great source of information on receptor expression is some recent work done by Pablo Engel's team, for which they looked at CD maps. And their first paper addressed CD1 through CD100 for human. But not just in blood, as we did in BD with the big databases we have, but they did it for thymus and they did it for tonsil too. And they looked for the corresponding expression of the number of ABC units or receptors on each one of those. And they looked also for where those CD1 through 100 are expressed on different cell types. Because it is that information, if we just look into the databases, it tells us what's on what type of cell and how much is expressed on each cell. Now that's all done in normal blood. So all bets are off, as we know, when we go to a disease process or an activation, things can change. But it's a starting point because it's great to know that a specific specificity may not be in a cell, a different cell, and that information helps us perhaps put the right color on the right specificity. Now, Pablo's team did a lot of different work, and I extracted this figure out because it told a great story. It looked at the expression of a number of specificities important in the different subsets of CD8. And as we look at this, it's a great source of information that says what's on what cell and what's not. What's on every cell. So we can see co-expression patterns for different subsets of cells. And it's our ability to place those colors then in the right places to be able to offset problems of spread. But if we look at something like CD45RA for the central memory versus the naive population, you can see where you might be able to use a color a little bit differently, and where something that might spread into another specificity like RO, we can put the right color in the right place. So this paper is a fundamental paper, and I think that everyone should have a copy and look at it and read it very carefully and look at the database of specificities. This team that built this paper actually is a sub-team out of our HLDA that has given us all of our CDs for human immunology. And it was their idea to expand on all those CDs and to give us more pertinent information about how to use them, especially in flow. Now, another place to get information, especially for all those biologists that work in the field of mouse immunology, is that I've worked with Hervé Luge at CIF in Marseille, who works in a program with Bernard Mallison and Marie. And we decided to look at the expression in spleen of mice to be able to help give lots of information as to the expression profiles, and then how we translate that information into panel design. So I just took a snapshot of one little piece of data. But in the mouse world, we could do something very, very different. So in this field, we looked at basal expression. We stimulated with anti-CD3, and then we also injected those mice with poly-IC and looked at an inflammatory response. And we can see for all of the different receptors that we did, almost 300 of them, we can look to see for what it was up and down or down regulated. And it's almost the very first time that you get a composite look at all those potential activation markers that you might never have known they were going to move. Those specificities that are listed in this deep database will allow you then to take that information and build it into a rational design for your panels. Hervé and I are working on this for publication. We certainly have a sense of urgency to get this out, and it was nice to see the new data that Hervé just sent over just last week to actually place in here. This is a live database, though, and I think it will be, available out there so that individuals can just look in there and see what those expression profiles may be on multiple different subsets of cells. So there's tools out there and data that help us know what's on what cell and how much has been expressed. Now, another area of focus is on the choices we have for the colors. And I borrowed this slide from Mike Stadniski and from Mario. And it's been a slide that we've been using for maybe the last 6 or 7 years to tell the story. It was designed first by Mario to put a concept out there to say, when those very first brilliant violet dyes first came out, the best dyes we had were PE and APC, and for many of us here today, we remember the importance of those two. We save them for the very special spots of our panels. But today, APC and PE are kind of average colors now because of all of the different development programs that have occurred in this world. So when we look at this continuum over time for the different colors that have been developed, we see that rich time where a lot of those Cerogen dyes, the brilliant violets and the brilliant UVs, and then also that introduction of the brilliant blue dyes from BD in that space. And then today we are seeing the Novaflor colors come in. So for everyone in our field, this is an amazing time to have so many choices. The colors are getting better. They spill less. They're very bright. Their extinction coefficients are in the millions. And with those, we can do many things we couldn't do before. So it's not always that we need a new instrument to take care and use these. And it's always easy to have an instrument that may have 30, 40 different colors and detectors on there, whether it's spectral or whether it's conventional flow. What is important is to try to define and see what are the best colors to use, and for many of our flow cytometers, we just need to refilter them in some cases and take out some of those older filters, because many of these new ones are replacing the old colors. And many of them are actually new, with new filter sets that are needed. So it's a combination thereof of taking, older technology sometimes and refiltering versus moving into new technologies that you can do everything with. And that's a challenge for all of us, but just because the age of a flow cytometer in your lab, it doesn't mean you can't use these great new dyes and conjugates that are being created today. If we take a step, though, and look at a laser line, there's a story that sometimes is overlooked, because for the most part, when we look at a laser line, we just look for all the colors, we can kind of see where they spill into each other, and we just move from there to our panel. But there's no need to stop there. So what I'd like to do is take a laser line apart for you a little bit. Every laser line has a story. The story shows us where cross-laser impacts are. There's also impacts on spread into the detectors on the same laser. So what I've seen over the last 5 years since the introduction of the high-parameter BD-FAX Symphony technology, we began to look at colors and interactions and decisions in a completely different way. So let's take a look at the fact that understanding the dynamics of each laser line is important. And this experiment that I'm showing you really only needs to be done one time on any flow cytometer, because they're all different. And the value of this type of analysis can help you in your panel design. It's a fundamental. It's foundational. So let's take a look at the UV line itself. So this is an experiment for which, on any instrument, we would open up every channel and simply run a color and collect the information for every single detector that's there. So in this experiment, we use CD4 and we use blood. We conjugated the antibody with BUV395, which is the first color, off of that laser. And what we see here is a separation, a really good stain index separation between the negative population and the positive. And when it's punctated on the positive, that's really good. But when it spreads laterally, well, that could be a problem. So as you look at this, if you open up every channel, there's very little spread into any detectors, a little tiny bit into two of the detectors on the red line. But for the most part, that is a really good color. Now, if we move to the BUV 615, which is right in the middle of the laser line, well, we can see its problems. We can see spread in many different detectors in different laser lines that are there. And all of this information is really critically important, because you'd say to yourself, well, where do I use this color? Because there's so much spread here. Now, we'll go through some of those options in a little while, but when I look at a color like this, I think, let's go to a place where there's fewer receptors on the surface, because this is what it looks like when it's CD4, when there's over 40,000 receptors, not where there's 500 or 1,000. And we're going to look at those experiments in just a few minutes too. As we move to the end of the laser line, the BUV 805, everything looks good again. So it's like a Gaussian curve across that laser line. In the shorter wavelengths, it looks pretty clear. In the longer wavelengths, it looks really good too. Our problem, as we all know, is in that 600 range. So the colors at the end of every laser look and can be used very effectively in panel design. Something that lots of individuals don't pay a lot of attention to, but there is a story for each individual laser line. Many years ago, David Parks gave us something incredibly useful, a way to let us examine the differences between the colors, between the brightest to the least bright. And there's stories therein of how we use those. And we all know some of those, which is, if there's lots of receptors, well, we don't tend to use one that's really bright. And if there's very few receptors, let's try to use a color that is much brighter than others. But that only goes so far, because you still have to be careful with spread. But if we know something about that receptor density, it does help us. And it's one area that if we pay a lot of attention to, we can correct panels if we know that information. Translate that stain index into figures like this. This was an experiment that was run on a BD-Fax Symphony. And it tells a story of colors. And there's many different things that we can look at here, but to point out just maybe one of them. The brightest of all these dyes on this instrument, and again, we should run this same type of experiment on every instrument we have. We only have to do it once. Unless we go and change a PMT on a conventional box or a laser, then we can go back and recheck. But if we just run it once, we learn about that instrument. We can compare a sorter to an analyzer to see how easy we can move a panel from instrument A to instrument B. But here, when we look at the relative stain index for CD4, knowing the colors helps us in our panel design. Let's just take one example, and that is FITC. And that's way down at the end in terms of its efficiency and brightness for CD4. We're still able to see the difference between them. But the same color in that same wavelength, but made a different way with the technology from BD, is that brilliant blue is much brighter in the same exact channel. Now, it doesn't mean that we just need to replace FITC, because FITC is a fantastic color. It works inside the cell. It's something that has been conjugated to so many specificities that we can find a place for it. But in your panels, if you had many specificities where there's many different low expressed, well, there's an option for you to use in that same channel, not FITC, but Brilliant Blue 515. But also, there is a caveat about that color. It's not bright on every specificity. So learning about every color is also foundational for everything. That one color is very different than most every color that we've ever used in flow because it's not consistent. But in CD4, you can see the difference between that. Understanding the instrument, though, is also a really important thing, as I noted before. And in this table, it represents 3 different instruments in our field. You can note that some channels are very consistent between the instrumentation. But there are unique ones in here that are very different. Therein lies, it is difficult sometimes to take an existing panel from one type of technology and to move it over to the other. Because if the stain indices are considerably different, the pairing of the antibody to the color can have a major impact on how we look at our data. So it's always advisable to design the panel for a specific box. And if you're lucky, there is an instrument on the analytical side that has a pair to a sorter. Because when you do that, it saves the time of having to rebuild a panel on a different sorter. So paying attention to that is really, really important if sorting or functional assessments are important in the work that you do too. Our famous, spread matrix. A spread matrix that when I look at this, I tend to discount it a little bit. I would rather look at the data that you saw earlier in those slides on the BUV line. I can see it. I can see the impact. And we're going to talk a little bit about it in a minute. But when we build a spread matrix like this, and let's say it's all built on CD4, well, guess what? That's not what our panel is going to do, because not every specificity has 40,000 plus receptors. But what we can see by the color coding is that when it's red, there's a lot of spread, and when it's green, it looks really good. But remember, antigen density has an impact here. And thus, when you look at this, you have to think about what it would be relative to the specificity you chose, and then think about what that impact might be from a spread matrix. And again, as I noted, we're going to look at some of those experiments to help guide you to being able to take this type of data and superimpose it onto the panel you're designing. Remember, the greatest spread matrix is going to be the one where you have a well-balanced panel, and then when you put it into a spread matrix, it should look real green with very few problems. But this is usually where we start when we take something like CD4 or CD8 in every color and we develop these. It's good for some things, but if it's interpreted too much, then it can give you false perspective as to really what the spread is for any given specificity. Set of slides is going to deal with fluorescent spillover and how it introduces background and spread. And these concepts inside are going to go back to that spread matrix that you saw and enlighten all of us with where the true impacts are and where they're not. The first one I'd like to share is an experiment that Annette Meyer did in our BD program in Europe. And following a day where we worked, couple days we worked with all our application team. And that surprised me a couple days later when I got back to the United States with some very interesting data. So what she did is she looked at different colors and how, and this is CD4 again, and how they spread into the APC channel. So if we look at the PE size 7, well, that's real clean. And so is BUV 563. But as we move more in that 600 range, we begin to see a little bit of spread, all the way to a lot of spread. Now, please note on this that there are some prototype dyes that are also inside this deck. And those colors are available for everyone, irrespective of the type of instruments that we have. They're colors that we all need to be able to do great science. So what Annette did here is she started to work with dilution of the CD4. So let me share the next slide. So what is depicted here is that spread can be reduced with lower expression of a receptor or a bound receptor to a color. So again, this is CD4, and it's the 660 brilliant blue into the APC channel. And the blue represents a saturation of the receptor. And what Annette did is she simply diluted the CD4, and what we see is a requisite reduction in spread. And if we reduce and dilute it again into the green, we can see that the spread is reduced even more. But yet, we can still resolve that population. So here is an example of where if it's just a gating population of a lineage, we might not need that 5 logs. But if we dilute it, then the impact is that we get less spread, and that can impact other parts of our panel. Now, we can't do this with everyone because we do need saturation, because many receptors are upregulated, and we need the maximum separation because maybe the resolution is not that big. But this is a critical component here of understanding the impact of the expression number of receptors to a color. Spread, though. So here's a good example between two, the CD4PE, lots of 40,000 plus receptors, that with great resolution, right, we're able to see the spread into the per CP site 5.5 channel. But yet if we take Per-CP Site 5.5 into the PE channel, it's relatively nothing at all, although the resolution is less. So there are strategies that we can use to minimize the impact of spread. In this case, it's about receptor number and the choice of the fluorochrome that we do. So again, why would we waste a PE with so many receptors on PE? Well, we could take something like Per-CP Site 5.5 into the PE channel, but we also know something about Per-CP Site 5.5. It can spill into lots of others too. So it's multifaceted in many ways. As a function, though, of antigen density, it has lots of impact. So let's look at this one. Here's the same fluorochrome, CD4 or CD95, vast difference in the numbers of receptors into the Per-CP Site 5.5 channel. And we see that spread is significant when you have thousands of receptors, but when we move to something like CD95, it works really well. So if you think back to your spread matrix, and you saw the spread from CD4 that would be red, it might lead you to the fact that maybe you can't use that color or a specificity on there. Well, you can. You just have to find the receptor that is expressed much less. So it's not a bad channel, only if you have a lot of receptors. Spread as a function of fluorochrome brightness is also something critical to understand. So Brilliant Blue 700 is a new dye in the same channel as Per-CP Site 5.5. And believe me, it is very, very bright. And when we made it, well, it spreads everywhere too, just like Per-CP. But since it's so bright, we can use it different. So if we pick CD4, you can see the resolution is significant there. The spread in the APC channel is very, very big as compared with Per-CP Site 5.5. So it's about the brightness here. But if we wanted to lower the brightness, as we saw in the other experiment, we could simply dilute that and get the same impact here. So again, another fundamental of how we look at spread, and again, how we look at it against the spread matrix. Spread matrix tells us one thing only about the numbers of receptors that we used at that time in that experiment. Now, antibody dilution is another thing that we look at. So from here, we use a saturating concentration at about 1 microgram per test at a constant voltage in a conventional flow cytometer, and then dilute that down. And you can see that there is an impact on spread. But there's also an impact on resolution. So choosing the right one in there is just an example of how we might use that specific color and concentration. Again, when we're looking at things like CD8 and CD4, those primary receptors that are gating ones, we can use these colors in different ways than we can for the many other receptors that are buried in our very large cocktails, if we choose to in this way. And in another way too, it's a way to use less antibody, especially in those big lineage markers. But always be careful. Voltage titration is certainly something that we get lots of questions on all the time, and where and what its impact is on resolution and spread. Again, what you can see here is that as we change the voltage, there is a reduction in resolution. But changing the voltage doesn't really change the spread. Again, a fundamental, and individuals try it sometimes on their flow cytometers, but there's not a drastic reduction here. So we kind of get a double negative impact because we just lowered the voltage, but we reduced the resolution, and thus the separation between a positive and negative can be more than what we really wanted. Move away from the spread. There's also a series of things that we all know. They're reminders, but they're important fundamentals in what we do in panel design, because not all panels we work with are just on the surface. And the complexity rises when we go inside the cell. And there's a lot of learnings, as we noted some of them at the beginning of this presentation. So those are that not all colors work in intracellular applications. This is always confusing. And in fact, not all colors work, not all antibodies work, not all clones. And for many of us that worked in that field of phosphorylation work, CD19 was always a problem, but it was every clone was impacted by the fixation processes. We have to understand panel design bias here, because when we pre-select colors, such as a PE that works well in intracellular and a FITC, then we take those out of where we might like to use them on the surface of a cell, but we can't use them there. So there's bias in there. And it challenges us in our panel design, because we just don't have all the potential colors that we do when we work on the surface. Now, one of those other areas that we have to pay a lot of attention to is, what kind of proteins are we looking at? Are these a lot of different types of intracellular phosphorylation proteins, or are they transcription factors, or are they cytokines themselves? And each one of those classes tend to have a unique type of a buffer system. In the phosphorylation world, we have everything from 100% methanol to a detergent that can work, because sometimes we have to get into the nucleus. But then that high methanol has an impact on colors. And today there are some big, databases, one at BD that you can go in and look at, that we did several years ago. And it shows not only the impact to each of the phosphorylated proteins, but also the impact on the cell surface receptors. So the selection of those clones for cell surface markers are equally as important when we're looking at intracellulars too. So it's about data and information. So whether it's in papers or whether it's in databases, what we really want to do is know if they work in a given buffer system or they don't before we start any application in intracellular proteins. Example of this came from the work that we do inside, where there's a rich, treasure chest of reagents, and we can look across many things to try to learn more about a specific color. In this experiment, we used Vax-Lice. We used an antibody, CD4, that could be used in different ways. We can stain, and then we can perm and fix, or we can perm and fix and then stain at the end. And as we look at these two colors that are in the same channel, look at the differences. So right off the bat with live cells here, with Vax-Lice, what we can see easily here is that the brilliant blue 700 is brighter. So there's some advantage, maybe not for CD4, but there's advantage for other specificities with fewer receptors. When we pre-stain, though, to the permeabilization step, the Percy-P is wiped out completely, but the brilliant blue maintains it. And then if we look to a post-perm stain, you can see a couple things here. You can see the stain index collapse a little bit on the CD4 with Percy-P because of the negative population getting a little broader and then moving to a different mean channel. But also on the Brilliant Blue 700, a little bit of change there because it did have an impact on the intensity of color. So again, knowing about these colors and their impacts are truly important to everything that we do. They're just part of the fundamental approach that we have to look at when we're doing panel design. Now, one of those steps where we have a lot of fun when we work with anyone, our programs internal or scientists outside, is we put a little pressure on everybody that we need to see a biological tree. What are you looking for? And in some cases, there's a lot of knowledge. In others, there's a lot of more help that's needed. Sometimes we build them. But let's look at an approach that we see quite common. And that is that we get. But that's all we need, is an idea concept about the flow of the specificities that you need. It helps us identify what's on one side of the tree, what's on the other side of the tree. It tells us a little bit about each specificity, so we can look into the databases and try to determine if they're primaries or secondaries or tertiaries, or how many receptors there are on each one. And when we put all those together, it gives us a place to start. We know how many low expressors. We know how many high expressors. If it's a broad panel, we know there's multiple lineages, so separate panels. And this is a great place to start. But unfortunately, not everyone does, and we're going to look at that at the very end. But we do like to do this because our goal at the end is to take something exactly as written this way and transform that into this, where we have defined populations. The orthogonal expressions and the specificities that we see look really good. The rich biology that we get out of it is all about how we balance the color to the right one, and how we looked at different types of cells to get that information from T cells or NK cells. So it's our opportunity here is to balance that, and we need that data of a biological tree, and once we have it, well, everything falls in place. So we go back to how we build a better panel, and we remember that slide at the very beginning where it shows the suboptimal panel, and it says, well, we could take those fundamentals that we just talked about and we can change a couple things and make a significant impact to the resolution of multiple different places in this panel. So let's take a look at a couple of them, not at great depth, but to show you the ease of using those fundamentals in making change and how it impacts and builds a better panel. So we both of them up. The upper one is the not so good panel, and down below it's much better. And you can see that separation from multiple ones. You can see in the MRAS how those populations just are pulled apart. We can see in the TSCM population significant changes here. But what's interesting as we look at here, it's the same colors in the panel, We never changed those. We changed something else that had an impact on those. The question is, like, what did we do to be able to get to that type of data? And we didn't change anything on that TSCM as an example. This work here was all done by Mirko, a scientist in our group, and I just think he did a magnificent job, he and his team, in bringing this data together. So the panel comparison that we have, when we really look at the TSCMs, you can see in the middle there the increase in numbers from about 2% up to 17% if you work on different parts of the panel, make a couple changes. So it should be noted in these two examples in this panel, everything was performed with the same donor and the samples were run on the same day. We just corrected one of the panels. But despite the use of that same fluorochrome for markers that we see for the CD95 and the CCR7, there's dramatic difference in detection and the quantification of those different subtypes. And it happened due to a simple switch of one fluorochrome matched to CD45RA from APC87 to BV480. Now, just thinking about that, the APC87 is not a very bright color on a receptor that has lots of different receptors, lots of different subsets. It's a secondary receptor. But when we put more color on it, we can spread that out. And when we do, we open the biology on the 45. And by virtue of just doing that, what we can see is the different levels of the CD45RA in the naive population that we couldn't see in the above panel because they're all clustered together too tight. But that small, subtle change allows us to see something considerably different. It's all about the balance of the fluorochrome into the right specificity. Simple changes sometimes, if you know how you built your panel, go back and look at the biology you used. So as we move forward in the last few slides here, there is a question that's a common one in this field about, where do we start a panel? And that's a hard one because individuals have all different ways to start a panel. Sometimes we have specific colors on specificities that we always want to see. A certain color on CD45 is a good example. And it's got to be the same in every one of my panels. Or I've got to have the same live-dead discriminator in every one of my panels. So these biases that we put in and approaches that we start impact everything downstream. So I'd like to go through a couple different approaches. They all came from scientists that are inside BD. It's no different than any place else in the world. We all look at this technology approach in different ways. Now, the first approach, though, that I'll share, to be all truthful, I probably wouldn't use it myself. I think you can get to the answer at the end. I think it is a common approach that are used by many. But as we go through it and think back of those fundamentals, maybe it'll make sense that this one might not be the most choice one to use. One of the scientists in our team, with all transparency, shared this one. So the first thing that the scientist did is they go to the catalog and they look for the availability of specific conjugates for all of their specificities, but they pay a lot of attention to some of the new specificities, right? Maybe they want to add a couple new specificities that just came out. And as you can imagine, you know what happens. We only have a couple colors. So what they do is they choose one of those colors because they don't want to make a custom conjugate of it. And thus they place a bias in there already. Well, they build their biological tree, and then what he said is, I go through and I look at all the markers that might be co-expressed on any given cell, and I match my gating strategy with those color selections, so that looks okay. And then they take a chance to look at the resolution and the spread for all those conjugates, but at the very end, you can see problems. So he juggles them around. He has to buy other reagents. He starts to rebuild the panel, and what's happened here is sometimes we have to purchase more reagents than we might need to here, but there's a lot of what-ifs in this panel design. And then if it just doesn't look like it's going to work, well, then he goes back to the second best choice of that original marker and then starts again. So it's a common approach. We take what's in our refrigerator sometimes and we go from there. And we take an old panel and we just try to add to it. But lots of times it's better to take a white piece of paper and use other approaches. So a second approach that's used, and it's a very interesting approach, is that we start with the bright format fluorochromes. And those bright ones give us a chance to reveal biology from those dim markers. And from that setup, then we go to the moderate ones. Now the moderate colors are circled. So we go through this process back and forth to get to a point where finally we start to look at co-expression patterns. And from there we can pick any color like 605, and we realize that it spills into an adjacent color. Those could be 650 or BV711. Realize that it also spills into PECF 594 due to cross-laser excitation. Well, then we tend to move those around a little bit to find the best combination. And that approach works exceptionally well. A third of though is one that I first saw from a scientist in Australia in our team, Andrew Lim. And when I first saw it, I just kind of sat back and I thought to myself, wow, this one makes a lot of sense. And he works with fundamentals all the time, and he's the scientist that never leaves out of control. So when I look at this, the first step that he does is he breaks all the laser lines by the wavelength down. And he puts them into 3 different sets, from the blue-greens, the short wavelengths, to the near-infrareds, to that central zone that's a lot of trouble. Now, for everyone on this line, in all the many panels that I've made in the higher dimensional space, I use what Andrew shared with me. And I see great consistency in panel design. I'm not saying it's better than others, but it makes a lot of sense to me with the fundamentals that we have here. So what he does first is he begins placing the long wavelengths on the specificities at the top of the biological tree. So he takes advantage of all of those at the very end of the laser lines. Then what he does is he moves to the middle of that panel with the short wavelengths. Again, really good colors, still medium to high expression. So we're not going to have a lot of trouble. And what he does at the end is that he saves the specificities that are low expressed for that valley of doom that we all know by looking at our spread matrix and the real data, where there is a significant amount of spread when we look for CD4s, but when we look for many of these markers in deep panels, well, there's not enough receptors there to actually cause a lot of trouble. So you can see the rationale of all three approaches. They can get you there. The one takeaway here is the fundamentals, if used inside, can always make you a better panel. Now, the approach that you start with is always up to you. And again, there are great panel designers out there in different companies and different universities, pharmas, and biotech, but they probably do use some of those fundamentals to get to a great panel. So I'd like to program with a few bullet points and things to consider, and if we take them home, there might be a great step forward for many of us on the line, that there are many ways to build these panels. Fundamentals and the foundations of each one of them, if you fill it up inside with all of those, chances are your panel is going to be better than the last one you built. There are lots of different factors that contribute to this. And those are part of biology. They're part of those fluorochromes that are being made even better today. The instruments that we use, whether they're conventional or spectral, all of them, all of these fundamentals are important. And then we get to that protocol because, again, protocols bring bias into what we do. But overlooking all these factors can have an impact. And that impact is something that should be of the greatest value to you. Most of these fundamentals that I've talked about today here, though, have everything to do with an analytical panel. Because at the end of the day, on a sorting panel, if you don't port that analytical one over to a sorter, it's just about isolating that one population or two or six that you need, and you might go about the panel design in a little different way. But it's all about what you're trying to achieve. Now, how we select all of those fluorochromes, how we understand and look at what that spillover is between lasers and where it spreads in is certainly something that if we do it right, the data gets better. You saw those examples of choosing the right color for the biology. And then at the end, if we pair all those fundamentals that we have in flow with those biological trees right up front, it becomes even easier because we can see what's on one side of an arm of a panel and what's on the other side. Remember, they don't cross over. So if we use these fundamentals, we have a greater chance, a greater opportunity to be able to build a really good biological panel with great data. And when we do that, then our experiments give us information that we've never seen before. So in conclusion today, I'd like to acknowledge a number of individuals. So many at BD that I can't even put them all on 1 or 2 or 10 slides over the last 20 plus years I've been with BD. For the countless discussions and for the individuals that contributed to this in so many different ways, from individuals that worked in FlowJo to individuals that are the physicists in our business, to the individuals that built the reagents and the instruments, I thank them. And for the insights of many of those on this list right here, for all the efforts that they put in to help you and to bring ideas and concepts back so it makes us better. I I thank all of them. Specifically, though, to Hervé as a friend, as a contributor, as a colleague in many ways, I thank him for all the work that we've done together on the mouse antigen density program. And also to Mike Stedniski, who is pushing new colors into a field in a new company, and to all of the work of Mario's laboratory that has brought so much to all of us in our field, and also that of the efforts of Steve Perfetto and others in their laboratory over time. I thank them too. So from that perspective, I'd like to move this presentation over to Manisha, who can help organize and get the questions in queue so that we can address those for you. I thank you very much.
Thank you, Bob. Thank you for a great presentation. Always learn something new hearing your talk. So appreciate it. Thank you. I enjoyed it, and so I hope so did our audience. So with Bob, I would also like to introduce the audience to Ravi Hungurani, who is our application scientist, who will be available to in this moderation session to answer any questions. Thank you for all your great questions. One of the questions that we have is from Ravindra B. from Intest Biopharma, where he's asking question, Bob, on with the emergence of spectral cytometry. He wants to understand if there will be, it will bring significant changes in the world of conventional flow cytometry, and also what will be the impact. Bob.
Thanks, Manisha, and thank you, everyone. You know, I think it's a really good question, a timely question today with the changes that we're seeing in the field today with new technologies. And you know, it's not the first time we saw changes. In around year 2000, we went from analog technology to digital, and it revolutionized the way that we were able to do flow. And as we've seen over the last few years, there have been a number of advancements. The advancements that Yongchen had done with the instrument that's in Beckman Coulter today with the cytometry technology, and that was a significant impact to how we look at flow, even in conventional space. And as we've transitioned over and saw even new spectral instruments, it's great to see it. And so the question is really good about, you know, is it going to change our field? And I'd say in many ways, yes, it is. It brings great advantages to us in some cases of ease of use, being able to break apart colors that are very close to each other. And I think this is a major contribution. But I think in some ways, the spectral has helped us, I think, a little bit with looking at many of the old colors, things that are close, and actually colors that probably we won't even use in the future. We can break apart colors that are V450 and C500 and PAC blue and BV421, but honestly, most of those colors, or many of those, aren't very good colors anymore. And I think with the advancement of colors and how we separate them through the spectrum, with some of the very new scaffolds that are being used out there today, along with all the Sirigen technologies that are there, we're getting better and better colors, ones with higher efficiencies, and they're helping us kind of open the biology up a little bit. But when we get back and we look at spectral, I think yes, there's great things, but we have to learn to use that technology. It's not just a transition over and you use it. How we control our experiments for each one of them. We know how to do that in conventional flow. And some of those things are going to take us, time. And even within our own company and what we've been working on in spectral space at BD, we're learning inside and making sure of every different dimension, because one clear thing that we know we need in this field is that with the transition of spectral, it helps us in that analytical way in many ways. But what we need to do in many of our laboratories is to pair instruments across to make sure that we've got the ability to take a panel and move that into a sorter. And we need it today. We need to do that. And I think that the positions that we have out there as new technologies come, we're determining how to do that. We're going to have to work with the speed of spectral and mixing and the quality of that to help us with those events that come through. So I think just to bring that question back, I think that spectral is a great way. I think that conventional flow is really, really good too. It's about what we see with biology. And the best biology is that separation between a negative and positive across as many, and how its ease of use as we get higher and higher in the number of specificities we check. But, the whole field of flow is not up there at 28, 30, 35, 40 colors at all. The majority of flow is considerably less that we use in our laboratories today. So I think it's something that takes time to transition through. And I think with that, it's going to change. It's going to take certain steps to change to make sure that we build even the correct reagents for the type of devices that we have, how we build tandems and how they're seen in spectral. All of that needs to be done before we're going to be able to say that there is a true new technology platform that is going to change everything we do in flow cytometry today.
Oh, thank you, Bob. Thank you so much. Ravi, did you have anything to add, or should I move on to the next question that we have from Alexandro at UCLA?
Thanks, Manisha. Just wanted to piggyback on what Bob said and said, in the end, the key concept over here we're talking about is the resolution. So selecting the appropriate color, having a few options of having bright markers in the same channel, might not resolve, or the dim markers might not be able to resolve the specificity of your interest. So having that key concept about dim antigens being on the brighter formats still remains the same in across platforms.
Understood. Thank you, Ravi. Thank you, Bob. So the next question we have is from Alexandro at UCLA, and also Karina from Case Western has a similar question. So I've tried to club the two of them together. So both of them want to understand what it means, Bob, that when you say the color at the end of the laser, what does that exactly mean? And Karina wants to understand how would we know which is the first color for the laser. So Any tips and tricks for them?
Okay. If we can, let's advance back if everybody can see. I think it's slide 13 or 14 in this deck. And here it is on 13. Let's see if we can both. Can the audience see this?
I think they're working on it. Why don't you start by answering verbally, Bob, while they're trying to get to that?
So across every laser line we have, there's a series of colors that go from the very smaller wavelengths on any given laser to the wavelengths that are longer. And on slide 13 it's an example of the UV laser that starts with the colors that we've selected to place in here, which are the brilliant UV reagents. They start with brilliant UV 395. And that brilliant UV 395 is a base polymer that we use. That's considered that first color with the smaller wavelength. And as we transition through BUV 496 to BUV 563, 615, BUV 661, BUV 737, we come to the last one that we've used up to this point, which is the BUV 805. So it is that specific color that we see at the end of the last color on that laser line. Now, what's interesting to note here for many people on here, and the question does come up, is that when we look at all those different tandems that are inside there, individuals always ask whether or not it's always paired with the same base polymer. And the answer on that laser line is they're not. So we can make a multiplicity of base polymers, and to choose the appropriate base polymer that would best go with an acceptor molecule to make the greatest color. So although you see some spill and with the spectral viewer as we see here, when we go back to the other data, the real data, as we saw in slides 15 and 16 and 17, the real data there shows where that spread is and the spill over to other channels. So when we look at a laser line, there is a story where at the beginning of a laser, there's very little problem. At the end of a laser line with for instance, BUV 805 here, there's fewer problems. The problems occur in the middle, across laser excitation, as well as between the tandems themselves.
Thank you, Bob. That's a good explanation. So, and if for others, in case you have any more questions, just feel free to reach out to any of us, and we would be happy to kind of, explain in detail or have contact our application, right? So, another question that came up, and I think that would kind of, benefit everyone, and it seems like it's again from Alessandro at UCLA. He's asking us about how are we defining the secondary and tertiary markers? How would one go about doing that? That kind of dovetail nicely into the biological tree, Bob and Ravi.
As I pointed out earlier on one of the earlier slides, the publication that Yolanda Menke and Mario wrote several years ago was the foundation for it. But really what they said was that the markers, the primary markers, are those ones that are easily discernible between the negative and the positive, and which are highly expressed. And many of those are those early markers that we saw, the lineage markers, the CD4s and 8s and 19s. Now, how we define a secondary marker based on that publication was that there is a continuum of expression. So when we look at a specificity like CD45RA and look at that expression profile with virtually any color, let's say a bright color, we'll see that stretch across 4 or 5 logs in conventional flow cytometry. Now, I've had a slide that I've used with, in a number of decks over time, and it's a wonderful slide that was built because what we did is a larger panel, and then we backgated in. And what we see in that continuum is that there are many different types of CD45RA. And that continuum is made up of a number of individual peaks. So what's important in flow sometimes is to spread that continuum marker out as far as possible because the biology gets spread. And if you don't use enough color, then it's all compressed into one place, and really there are different subpopulations there. Now, the tertiary markers are different. These tertiary markers are oftentimes what Mario had done earlier, is that he had defined them as being very small numbers of receptors per cell. Very, very small numbers. And we see that, like the CD132s, we see markers that are around 100 receptors or 50 or 80 receptors. And then along with that are markers such as that are activation markers that have, over time. And what's very interesting there is we can't always see them in the human because we can't do certain experiments with human. But when the work that Hervé and I did and his team did on the mouse, we actually did a lot of different activation protocols. And inside that deck is going to show everyone with the many different types of markers that actually move when there's an inflammatory response or an activation that's there. And on that slide, although I moved through it quickly, which is just one snapshot of just thousands and thousands of data points, you can see the upregulation of H2. You can see lots of different markers that move. So I think a little bit of this new data, when we actually look at so many markers at once, we may end up reclassifying certain markers then from where they were as secondaries and moving into tertiaries. So I think a lot of it is in that publication, but I think the new databases that will be available and the new publications that come out are going to demonstrate much easier which specific markers, are up or down regulated perhaps as tertiary ones. But just remember, these databases are rich, and what we see with a receptor on one type of cell, like a B cell, and what we see how it works on a T cell may be completely different, not just in the number of receptors that we see, but in terms of what the function of that receptor is on that specific cell. And thus, these databases that have a lot of information about what's on what subset of cells, not just what receptors on a T cell, because there's different subsets. All these things, can contribute to understanding and making a better panel.
Thank you, Bob. Ravi, did you have anything that you would like to add to what Bob mentioned just now to this question?
I think just one note would be that, yes, Bob did mention this about the activation. Many times we are looking at activation, in vitro activation, and that might change the type of expression altogether. And many times it is always good to have a good titer for that activation model system. And many times we get this question that's saying that, what is the appropriate amount of expression for that model system, and it depends on what the activation system is. So be aware of the fact that the markers may change, and depending on that, at that time spot when you're doing the activation, that you need to make the appropriate choices for that system.
Oh, thank you, Ravi. Thank you. And with that, I would like to say that I'm sorry you all had great questions, and we will address them via email. We can't get to each of the questions that you sent us, but be sure we will definitely respond to them. So thanks so much, everyone, for your engagement and joining us today in our webinar session. We appreciate it. And also, just to let you guys know, this will be available as a recorded session on the BD Biosciences webpage, as well as the YouTube channel. Again, also, we are excited to let you know that we have a series of Harmony Talks planned for the future, which we hope you will be able to join us. So with that said, have a great day, everyone. Thank you again for joining us. And Bob, thank you so much. And Ravi as well. Thank you.